Hello everyone. So now we are going to discuss about plasma membrane, which is once again a part of biochemistry 1.1, subcellular organelles and cell membrane. Now below is given my YouTube channel link which you can refer to for your lectures, and if you find it difficult to access this link, then you can just type Dr. Bijoya Chatterjee on YouTube and you will find my channel. So we'll begin with the functions of plasma membrane. Now plasma membranes are also known as fence with gates. So if you imagine a big college campus which has a gate. Now, so the whole boundary of the campus is known, you can imagine, as a cell membrane, and the gates are uh, the ones that are allowing uh, ions and material to move in and out of the cell. The cell membrane's job is to control what is going in and out of the cell, and it also receives signals. So here is a diagram which will make it clear for you. Now the plasma membrane is selectively permeable. It transports ions and molecules. Receptors for biomolecules like hormones, neurotransmitters are also present on plasma membrane. Plasma membrane enables compartmentalization, that is, it separates one cell from the other, and various cell organelles are separated by membranes which are lying within the cytoplasm. Cellular processes, example cell addition, ion conductivity, and cell signaling, are also carried out by plasma membrane. Plasma membrane enables cell motility during cell division. Now let us look at the composition of plasma membrane. It is composed of various macromolecules, and these include lipids, approximately 50%, proteins 50%, carbohydrates 2 to 8%. The carbohydrates include uh, glycoproteins and glycolipids, and there is also a small amount of cholesterol present in the plasma membrane. Now we come to the lipid bilayer and the various lipids that we see in the plasma membrane. Now this lipid bilayer or the plasma membrane is actually a bilayer, which means there are two layers of uh, phospholipids, and each layer is of 25 angstrom approximately. This may vary, and therefore the both the layers together form a thickness of around 50 to 80 angstrom. Okay, so now let us see the details about the phospholipid bilayer, since it is the chief lipid present in the plasma membrane. Now this phospholipid bilayer is made up of phospholipids, which has a globular head and tails. Um, so on the cytosolic or on the aqueous medium, the head side is facing, whether it is cytoplasm or extracellular fluid, the head part of the phospholipid faces the aqueous medium, and this forms the hydrophilic region on the extracellular and the intracellular part. The central part is known as the hydrophobic core, and this you can imagine like two pieces of bread with butter inside. So the slices of bread on each side are the hydrophilic heads, and the butter in between is the hydrophobic tails. So the phospholipid bilayer is like a bread and butter sandwich. Now the phospholipids that are mainly present in the external layer are choline containing phospholipids, and ethanolamine and serine containing phospholipids are found in the inner layer. This model was proposed by Singer and Nicholson, and it is known as the fluid mosaic model, and it was proposed in 1972. Now phospholipids The simplest phospholipid is phosphatidic acid. The other examples include phosphatidylserine, phosphatidylinositol, cardiolipin, etc. Let us see the structure of phospholipid. It is basically a glycerol molecule. Now to the hydroxy group of each present on each carbon is esterified with various acids. For example, the first carbon is esterified with phosphate group, you can see here, and the hydroxy group present on second and third carbon form an ester link with fatty acids. So fatty acids form the tail region, and this is the hydrophobic region, whereas the hydrophilic region is composed of the glycerol head. So this is the glycerol head, and there's also phosphate present. These glycerol and phosphate are the hydrophilic head region. And the tail region contains two fatty acid chains. The fatty acids may be saturated and unsaturated. Because of unsaturation, there is a slight angle in the chain, and this is known as the kink. You can see one double bond here. Now this lipid bilayer shows free lateral movement, and this lateral movement is known as membrane fluidity. There is some amount of cholesterol that is present in the phospholipid bilayer, and it plays a very important role, which we will discuss in the next slides. As you can see here, that the brown colored molecule here is the cholesterol, which is a rigid structure. The phospholipids are this purple colored structure with two tails, and the large blue colored um, molecules are the proteins. So we can see some amount of cholesterol present in the bilayer, in the phospholipid bilayer. The other modifications of the plasma membrane are known as lipid rafts and cavioli. Lipid rafts and cavioli are examples of cholesterol enriched microdomains in the cell membrane. Microdomains means very small region that are present in the cell membrane. This region of the cell membrane is very rich in cholesterol compared to the remaining cell membrane, and because of this richness of cholesterol and its difference in composition, lipid rafts can influence membrane fluidity and membrane protein trafficking. So now next is the fluidity of the membranes. So as you know that the membrane is fluid and it is semi-permeable. So what affects the fluidity of the membranes? So first is the cholesterol content that alters the fluidity of the membrane. The other component that alters the fluidity is the unsaturated cis fatty acids. If the unsaturated cis fatty acids generally increase the fluidity, and unsaturated trans fatty acids decrease the fluidity. Now let us see how cholesterol influences the fluidity of the membranes. When cholesterol concentration increases, the membrane fluidity decreases on hydrophilic core. So if cholesterol is present in more amounts in hydrophilic area, the membrane fluidity will decrease. And just the opposite will happen in the hydrophobic area, because cholesterol itself is hydrophobic in nature. So if it is present in hydrophobic area, fluidity will increase. Similarly, when the temperature is less than the melting temperature, cholesterol will decrease fluidity, and when the temperature is greater than Tm, the cholesterol will increase fluidity. So these are some factors which determine the fluidity of the membrane. Now we come to the next part, that is membrane proteins. Membrane proteins include integral membrane proteins, transmembrane proteins, and peripheral membrane proteins. The transmembrane proteins are also examples of integral membrane proteins, but I have taken it separately as it needs greater explanation. Now, integral membrane proteins are proteins that are deeply embedded in the bilayer, in the phospholipid bilayer. So, it is inside the phospholipid bilayer, that is why it is integral. They interact extensively with the phospholipids. They are attached by hydrophobic bonds or van der Waals forces. Um, some of the integral proteins span the whole lipid bilayer several times or more than once. So that is known as transmembrane proteins. Are the various proteins that are found in the phospholipid bilayer. So this is the peripheral protein, this is the integral protein, this is receptors, enzymes, transporters, ion channels. All these are different types of proteins that are present in the phospholipid bilayer, and these help in transportation of ions and molecules from one side to the other, and if they are enzymes they may also catalyze reactions. Now the transmembrane proteins are, as you can see here, this is the phospholipid bilayer and this is one protein. This whole thing is a single protein which is spanning the membrane, crossing this bilayer 12 times. So because it is crossing, the same protein is crossing the membrane more than once, it is known as a transmembrane protein. Transmembrane proteins generally serve as receptors for hormones and growth factors. They also serve as receptors for neurotransmitters, tissue specific antigens. Transmembrane proteins also form ion channels, and they may also be membrane based enzymes. The peripheral membrane proteins. These are the proteins that are present on the periphery of the plasma membrane, and either it is facing the extracellular fluid or it is facing the cytoplasm. And these contain ectoenzymes. Examples of such ectoenzymes present in peripheral membrane protein are 5' nucleotidase and alkaline phosphatase. These are present on the outer membrane. So now you can see this is yet another example where this is a peripheral membrane protein, this is a transmembrane protein, this is spanning the membrane only once and this is spanning the membrane thrice. This is the uh, peripheral membrane proteins, this, and these are the integral membrane proteins. This is also an integral membrane protein. Now lipid rafts and cavioli. I've already discussed what are lipid rafts. Lipid rafts are rich in cholesterol and they have GPI anchored proteins which are attached to the external surface of the plasma membrane at lipid rich microdomains. Specific role of lipid rafts and is endocytosis and receptor mediated signal. So this is the example, this is the diagram of lipid raft. You can see this is the plasma membrane. Now this is a microdomain, that is a small region which has some variation in which the cholesterol content is higher compared to the remaining plasma membrane. And there's also a lot of sphingolipid that is present in this area. Because of these two changes in fatty acids, there is GPI linked protein that is present here, and also caveolin is seen here, which we will discuss later. The second structure that is seen in the phospholipid layer is the caveoli, which is also a special type of lipid raft, and these are small indentations or invaginations of 5 to 100 nanometers of the plasma membrane, and because of this invagination it forms a flask shaped indentation on the areas of lipid raft. These are generally involved in membrane transport and signal transduction. Cavioli contain some special proteins, as you can see here, these pink colored proteins. They are known as caveolin. Now, LDL receptor and receptor mediated endocytosis of LDL. So this is the endoplasmic reticulum. Rough endoplasmic reticulum will synthesize receptor. This receptor molecule is sent to the Golgi apparatus, wherein it is packaged and it is transported to the plasma membrane, where it is incorporated in the plasma membrane with the help of this vesicle. Now, when the LDL particle approaches the plasma membrane, this receptor recognizes the LDL and it forms a coated pit, receptor coated pit, and then it pinches out, and then the lysosome fuses with this endosome, and then there is digestion of the cholesterol and amino acids present in the lipoprotein LDL. Once it is digested, these are released outside and the receptor again is sent back to the cell surface to recognize another LDL molecule. This is known as receptor mediated endocytosis of LDL. The carbohydrates that are present in plasma membrane are 2 to 8% by weight. They are associated with proteins or lipids. If the carbohydrate is associated with proteins, it is known as glycoproteins. If the carbohydrate is associated with lipids, it is known as glycolipids. Always towards the outer side, the N terminal on external surface carries carbohydrate, and the example is glycophorin. The plasma membrane, therefore, you can see that it is asymmetrical, which means the same type of lipids are not present on each side. There is an asymmetry. The composition varies on either side. There is irregular distribution of proteins. The carbohydrates are located on the external surface only, and special enzymes are located exclusively on the outer and inner side of the membrane. The specialized membrane structures include tight junctions, myelin sheath, microvilli, and membrane organelles. This is the diagram of tight junction, where two adjacent cells are very close to each other, and so uh, there are actually four membrane layers: 1, 2, 3, and 4. So sometimes they come so close together that there is a fusion in the second layer. So only three layers of membranes are remaining. That is 1, 2, and 3. And this fusion or this tight junction allows the exchange of material between two cells. And you can see that there are various proteins here, that is occludin, claudin, catenin, cadherins, and vinculins that are present. The cytoskeleton of the cell. Just like our body has a skeletal system, the cell also is maintained by a cytoskeleton. Although it appears fluid, still the cell needs a structure to hold it, and this holding of the cell is provided by the cytoskeleton. The cytoskeleton gives shape, motility, and it enables chromosomal movement during cell division. It is present underneath the plasma membrane. It is made up of microtubules and microfilaments, and these microtubules and microfilaments and the cytoskeleton contains proteins spectrin and ankyrin. So these are special proteins which are found in the cytoskeleton. Here you can see the cytoskeleton. This is the microfilament and these are the microtubules. Thank you. So hope you liked the lecture. Keep listening. I'll be posting many more lectures this year.